And our final speaker for today, Richard Sharp from the Cleveland Clinic.
Picked up on a subtle pause in Ed's introduction there. The final speaker, the final session, the last day of the conference. But indeed, I'm honored to be here and very much thank the host of this conference for the invitation to be here. I also wanted to acknowledge the support that my laboratory receives from many of the funders who are here. My laboratory at Cleveland Clinic receives support from the National Institutes of Health, specifically the Ethical, Legal, and Social Implications Program by the National Human Genome Research Institute, as well as the Human Microbiome Project and the NIH Common Fund. I have no research support that comes from industry and no financial disclosures to make. I did want to say a more personal thank you to three individuals who may be known to some of you in this group, but who really have been very consistent advocates for examining the broader societal issues associated with human microbiome research, and those are Drs. Lita Proctor, Jane Peterson, and Jean McEwen, all of whom work for the National Institutes of Health. So as the last speaker, I think it's really my job to be sure that I stick on point and get right at it. So I want to leave you with just two simple thoughts here as we think about some of these societal issues. And the first is one that grows out of one of the introductory comments and quotes that came from Julian Davies. And that's that if enthusiasm for microbiome research, the sort of enthusiasm that we're seeing at this meeting, is justified and supportable, then that message surely has to be communicated more effectively to the public. Why is it that if we were to walk down the street today and ask the next 100 people that we bumped into, have you even heard of the Human Microbiome Project, or have you even heard of metagenomic research? Almost none of those people, I think the odds are quite high, in fact, that not a single one of those people would know even the least bit about this new area of research, which again has the potential, I suspect, to be transformative of the ways in which we think about concepts of health and disease. In the US, it's certainly the case that the Human Microbiome Project is largely unknown. In our studies of patients, for example, patients who have chronic gastrointestinal diseases like IBS and IBD, we've never encountered a patient who had ever heard of this project prior to us introducing that concept. More than that, what we have learned is that most of what people do know about this area of research comes from various retailers. And so those manufacturers of antibacterial products, soaps and probiotics and things of that sort, these are the primary messengers with regard to public understandings of probiotics and metagenomic research and so forth, microbiome research and so forth. So what we did is we decided that we would do a small analysis, a very small pilot study, in which we asked what would happen if you went online to Google or one of these website tools and simply entered in the term probiotics and looked for additional information. And we looked at the first 100 websites that came up. And what we found was actually pretty striking, because we decided to compare those websites that were supported by a commercial firm, a firm that was actually selling a particular probiotic product, against those websites that were not marketing products. And what we found was that those that did allow individuals to purchase probiotics mentioned roughly 11 benefits on average during the course of that website to every one risk. By comparison, those government-supported sites and other websites mentions 3 benefits to every risk, an enormous difference. We also found that those websites were much more likely to endorse the use of probiotics for therapeutic purposes, and some of the specific claims that we found when we did this analysis was that probiotics could cure ovarian cancers, that they could be effective in treating autism spectrum disorders, and that they could help to prevent kidney and liver diseases. And a number, I could really go on, a number of remarkable claims about the therapeutic promise of probiotic products. And in my opinion, those statements are grossly misleading about the current status of the science. And it's not the case that we should be relying exclusively upon these companies to put out first messages about this area of research. Because those first messages can have a powerful effect in shaping subsequent attitudes about a new and emerging area of research like microbiome research. So in my opinion, and this is really the first point I want you to take away from my talk, this is a place where funding agencies really should be leading. And I'm particularly excited about the CIHR initiative that was mentioned earlier in this session, because there's great need for sustained public dialogue about the state of the science and the potential for microbiome research to really impact health and disease. The second point that I want to make is related to the gap that I see between the research that was described at this meeting, which was largely descriptive of microbiome populations, and so-called translational or interventional clinical studies, which I think really do represent that next generation of microbiome research. So in the United States, the FDA has adopted a fairly conservative stance with regard to those clinical trials. And in most cases, particularly when you're studying biological function or when you're studying the impact of something like a probiotic on a particular disease or a particular condition, a clinical outcome of some sort, you're going to be required, if it's an interventional study, to arrange for an investigational new drug application. And that concept of using an IND to conduct a clinical study is going to be something that many people that are coming from the microecology and microbacteriology worlds are going to find very foreign, I suspect. There's a great deal of cost associated with these types of applications and a great deal of process requirements and process burdens associated with that. So I think as you look ahead to this next stage in microbiome research and imagine what some of these really provocative and I think very important clinical translational studies might look like, it's important to keep in mind that this will come with enormous levels of regulatory burden in many cases. Even if you're taking a particular species, which is found in a common food, isolating that, concentrating it in a certain way, and then administering it within the context of a clinical trial, it's very likely that the FDA will require you to file an IND in that situation. Now, I don't know what the regulatory situation may look like in other countries, but for us in the United States, that's a great worry, that we now have all these tools to investigate the clinical impact of bacteria, commensal bacteria, and we may find that there is a major roadblock to conducting those important studies. Related to this point, in our studies of patients that have IBD, we found that many patients have concerns about manipulating their gut microbiota. And often they would see these types of studies that we would present to them as hypothetical studies through the lens of genetically modified foods. And they would express concerns that, in fact, tampering with one's gut microbiome might lead to irreversible effects, that it would be difficult to control those organisms once introduced into their bodies. They also expressed concerns about environmental release and the prospect that these new bugs might get out into the environment and wreak havoc in some unexpected way. So I think, again, this highlights, these two considerations together highlight this very large gulf, if you will, in terms of the social implications associated with the more descriptive studies, which have been the focal point for this meeting, and these clinical and translational studies that are still to come. And for me, of course, that does highlight the importance of being more sensitive to those sorts of ethical and societal implications. So finally, let me conclude by noting that I agree with our afternoon speaker yesterday that a wide range of microbiome research can raise a number of important ethical issues related to consent, return of results, and so forth. But those issues are really quite familiar for people who are specialists in research ethics and quite familiar for many of you in this room as well. I don't want to make light of the importance of those issues, but what I wanted to highlight here is that there are some larger issues that, in my opinion, are equally important to consider. Thank you. Merci.